So our first speaker this morning is Arthur Brown, who's a scientist and principal investigator at the Robarts Research since 1998, and has had a long-standing interest in neurodevelopment and regeneration. He did his postdoc at, his PhD with Janet Rossett, at the University of Toronto, and then went on to the Salk Institute in San Diego and worked with Greg Lemke. He then joined us here at the Robarts, and about 10 years ago, Arthur decided that he was going to direct his neurodevelopment slant towards spinal cord research. And with collaboration with Lynn Weaver, they've had a very successful and productive collaboration where they have been able to push forward the neurodevelopment slant of repair for spinal cord injury. He has won the Premier's Research Excellence Award, New Investigator Award from Heart and Stroke, is currently funded by the CIHR, and also enjoys funding from the Morton Cure Paralysis Foundation. So today, Arthur is going to give us a talk on the control of gene expression in the injured nervous system. So Arthur.
Just give me a second. Oh, cue me up. Sorry about this, and there I am. Thanks for the introduction, Mike. Today I'm going to tell you about a story that's really been developing in my lab for the last 6 or 7 years. And it stems from our interest in how genes are regulated, how gene expression is regulated in the injured spinal cord. And in a very simple way, after spinal cord injury, you can consider two types of genes. Genes whose expression goes on, which would result in increased nerve growth, in increased neuroplasticity, that is the rewiring of circuits that could underlie recovery. And then there's genes that would do just the opposite, would inhibit regeneration, inhibit nerve growth, and so limit, if you will, recovery. And we were interested early on in what controls these different types of genes. And if we understand what controls the genes, we might be able to manipulate that and affect a better outcome. So just by way of introduction, spinal cord injury is a devastating condition with huge socioeconomic effects, huge personal effects. For our purposes today, I just want to take a look at the scientific view of spinal cord injury. And here I'm showing you in a cartoon form an injury at this level. And the problem in spinal cord injury isn't so much the loss of neurons and glia at the site of injury. It really is the fact that the neurons in the spinal cord below the level of the injury are losing their normal regulation. They no longer receive communications from other spinal levels above the injury or from the brain. So it's really a problem of connectivity. And when you don't have the regulation of these neurons, that is when you get the symptoms that are well known, the loss of motor output, so paralysis, loss of sensory input, so lack of sensation, and perhaps more importantly, other problems that are less commonly dealt with, like chronic pain and muscle spasticity and autonomic dysfunction. So this is the problem that I'd like to consider today, and this is a photomicrograph that I got from a Stephen Davies paper, which shows the problem that we want to think about. This is an injured rat spinal cord. There's the level of the injury, spinal cord in longitudinal section. In green, they've marked the axons that are descending from the brain. And they no longer are able to connect to their targets below the lesion, and they retract back. And so the question we began with is, why don't they reconnect? They knew how to reconnect in the embryo, right? Because those connections formed. But after injury, one of the things that happens is shown here in red, that's GFAP staining, showing that there's an astrogliosis. The astrocytes proliferate around the injury, and they elaborate various molecules that can inhibit regeneration. And here we're talking about chondroitin sulfate proteoglycans that are actually stained here in blue. And so CSPGs, chondroitin sulfate proteoglycans, have been shown to be inhibitory to regeneration. Very simply, if you take neurons and try to grow them in culture in plastic dishes, if you give them a substrate of CSPGs, they fail to extend neurites. On the other hand, if you give them a substrate like laminin or fibronectin, they like those substrates, and you can get neurite growth. And if you alter the proportion of CSPGs and laminin, you can get varying amounts of growth. So others have shown that if you get rid of the CSPGs with an enzyme called chondroitinase, that digests the chondroitin sulfate side chains, and then you can actually get growth through the lesion with varying success. And we were interested in this from a sort of more of a developmental point of view. What's the pathways that regulate this? Why would these genes come on after injury if they're inhibitory? It's counterintuitive. And when we began to think about the problem, and we want to know how are these genes regulated, well, there's just too many CSPGs. It's a very large family of proteins. It contains neurocan, brevican, aggrecan, NG2, perlecan, etc. Large family of genes, and we couldn't really wrap our heads around the problem of studying each individual gene. But then we came across the idea of, instead of looking at the individual genes, let's look at the few enzymes that elaborate the chondroitin sulfate side chains on each of the CSPGs. So CSPGs all have the same structure. They have a core protein, which is different for each CSPG. And then on the core protein are the side chains, the inhibitory side chains, and they are elaborated by various enzymes, and those few enzymes are the same no matter what the CSPG is. So we decided to turn our attention towards just those enzymes to simplify the problem. So I'm showing you here kind of a horrible biochemical slide to show you that the first step in elaborating the chondroitin sulfate side chain is the addition of a xylose onto a serine in the core protein. That's the rate-limiting step, and that's done by the enzyme xylosyltransferase, of which there's two isoforms, xylosyltransferase 1 and xylosyltransferase 2, which I'll call XT1 and XT2. And then the side chain grows out from that with successive addition of sugars, and then the side chain is sulfated by an enzyme called either chondroitin 4 or chondroitin 6 sulfotransferase. So we just wanted to study how are these key enzymes regulated to get some insight into what's happening after spinal cord injury. And so we were in search of this regulator. Hopefully we would find sort of a master switch that would turn on sort of coordinately all these genes that are involved in CSPG biosynthesis, and our key was going to be to find the regulation of these biosynthetic enzymes. And so what we did, we took many strategies. I'm just going to show you one here. This is a bioinformatics approach, and we took the promoter regions from the three genes, from XT1, XT2, and C4ST, from three different species, human, rat, and mouse. And we used a software package from Genomatics to predict putative transcription factor binding sites. And we wanted to find sites that would be in common to all three genes, suggesting that it would coordinately regulate this battery of genes that make CSPGs. And we wanted to find it in all three species, suggesting that it was important enough that it would be conserved through evolution. And in fact, the program spit out a few different transcription factors that were in common to all 9 promoters. And we went on to study one in particular, Sox9. And the reason why Sox9 stood out to us is because of what was known about Sox9 in development. And one of the important roles of Sox9 is in chondrogenesis, cartilage. And in fact, Sox9 is known as a master switch for turning on matrix genes in cartilage. So particularly collagen 2A and various protoglycans require absolutely Sox9 activity to turn them on. And so it made perfect sense to us that a transcription factor that regulates the expression of matrix genes in cartilage may also regulate the expression of matrix genes in the injured spinal cord. So the first thing we had to do is say, well, is it there? Is it in the injured spinal cord? And so this is an injured rat spinal cord. This is the big cyst that develops in the cord, stained for GFAP and Sox9. I'm going to show you this in a high magnification in a moment. And then DAPI counterstaining so you can see the nuclei. And if I show you the high mag, you can see that there's lots of nice red nuclei appearing purple because of the overlay of red and blue. So that shows that there's Sox9 expression at the lesion, and in cells that are surrounded by green cytoplasm, indicating that they are GFAP positive. So astrocytes at the site of injury express Sox9. So this is really a new function for Sox9 in spinal cord injury. So it's there, but does it regulate these genes? So we turn to a cell culture system that we've used quite a bit now, which is primary astrocyte cultures. So we get primary astrocytes from the rat brain at P0, first day postnatal. And what we did is we drove Sox9 expression with a CMV construct. And that's the black bars here. And you can see that when we drive Sox9 expression in these cell cultures, you actually increase expression of XT1 over its baseline, XT2, and C4ST. So it looks like it's actually able to regulate these genes. Unfortunately, it doesn't do anything to laminin and fibronectin. Now, in other studies, we had shown that these cytokines, TGF-beta, IL-6, PDGF, are also able to upregulate the expression of XT1, XT2, and C4ST in culture. And so we thought, well, maybe the reason why those cytokines turn up those genes is because they first turn up the expression of Sox9. And so we looked in these cultures, and when you do that, we see that when you treat the cultures with these cytokines, you get increasing expression of Sox9. So, I don't know if I have mine. No. So what we think is happening is we're building a pathway. We're building a pathway in which inflammation is able to turn on Sox9 expression, and then Sox9 is able to turn on these genes that increases expression of CSPGs and is inhibitory to regeneration. This is just to show you now the loss of function experiment in which we take, I think we're losing that pointer. Let me try this one. Is it underneath, Greg? Yep. Yeah. Okay, so what we're doing here is we're using a Sox9 antisense siRNA, and when you use a Sox9 siRNA, you knock down Sox9 expression, and therefore you knock down XT1, XT2, and C4ST. And in fact, even if you treat the cultures with the cytokines that turn up these genes, if you use a Sox9 siRNA, you still block XT1, XT2, and C4ST. So the loss of function studies show you that. And in fact, now if you take a look, we got a pleasant surprise when we looked in the cultures for laminin and fibronectin, the sort of the good guys. And when you look at that, the siRNA actually resulted in increased expression of laminin and fibronectin. So at this point, we sort of saw Sox9 as pivotal, sort of able to determine the relative amounts, perhaps, of CSPGs at the bad guys, if you will, and laminin and fibronectin at the lesion. And so in our minds, Sox9 became a really attractive target for spinal cord injury, because one would predict that if you inhibit Sox9 or knock down its expression, you should result in reduced CSPGs and increased neuroplasticity. So how do you test this? We went to a conditional Sox9 knockout, which we got this mouse from Andreas Schadel, working in France. And the first thing I wanted to do was to just confirm the astrocyte culture system. We only used siRNAs. Now we can do it in astrocytes where we've knocked out Sox9. And when you knock out Sox9, we get about a 60–70% knockout. You reduce the Sox9 target genes. Now we can broaden what we're thinking about as a Sox9 target gene to include aggrecan, brevican, neurocan, not just the XT1, XT2, and C4ST. So these are the core proteins now, and it makes sense that they'll be coordinately regulated. Collagen goes down. Interestingly, GFAP goes down. So GFAP is really a marker of astrocyte activation. So it suggests that astrocytes are even going to be less activated in general, in addition to the effect on CSPGs in particular, but part of the program of Sox9 activation might be astrocyte activation. And then we wanted to try it in the animal. Does it make things better after spinal cord injury? So in this experimental paradigm, we gave tamoxifen, which induces the knockout, for 1 week. Then we allow a week for washout. And then we did a contusion injury in the mouse. And then we followed them for 14 weeks. Now we're looking at expression in the injured spinal cord. No longer are we in the artificial sort of astrocyte culture system, and you can see that in the knockouts relative to controls, there's a great reduction in Sox9 target genes, including the core proteins, including collagens 2A and 4A, including GFAP expression again. What happens behaviorally? Behaviorally, if we do the open field testing, you can see that the knockouts perform much better in open field testing using the Basso mouse scale of locomotor function relative to controls. They're doing better. They don't walk normally by any stretch, but they walk a whole lot better than the controls. And in fact, we had a feeling that the overall mobility was even much greater than this would suggest. So what we did is we put them in activity boxes. The activity boxes have infrared beams that monitor how much distance the animal covers in a given time unit. So over 2 hours, just to look at how much the animal moves. This is the Sox9 knockouts, and these are the control animals, spinal cord injured. And interestingly, here's the Sox9 spinal cord injured, and here's wild-type mice that have no injury. So they move as much as the ones that don't have injury. Just to show you a little bit of protein, up to now I've only shown you RNA, but in fact the protein is reduced in step with the RNA. You can see that GFAP is reduced. That's quantitated here. This is without about a 70% drop in Sox9. The knockout strategy is not 100% efficient. So you get GFAP levels coming down and CSPG expression coming down. And if you look in the, wow, that's kind of hard to see in this room, but this is looking at CSPG expression in the controls and in the Sox9s. Knockouts, and you'll have to take my word for now. There we go. Thank you for that. You can see that the expression here of the CSPGs at the lesion site and almost absent from the knockouts. And if we do it in cross-section and we look at the epicenter of the lesion or rostral or caudal, this is the normals, the controls, lots of brown staining for CSPGs, and very little in the knockouts. Okay. But what do we think is happening? So we're still working on the exact mechanism. But this is sort of a clue to it, and this is looking at neurofilament staining. And when you look at the lesion epicenter, one of the things that you don't see is you don't see increased neurofilament at the epicenter, which suggests that there isn't more growth through the lesion. So as I've sort of painted it, you'd expect decreased CSPGs at the lesion epicenter, and therefore you should see lots of regeneration going through the lesion. We don't really see any evidence of that. I'm not saying it's not occurring, but here it's suggestive that it doesn't. But in fact, if you look rostral or caudal, there's much more neurofilament staining. You can quantitate that here in the knockouts. And so I just want to give you an idea of what we're thinking. We're thinking about it in this sort of way, that this is WFA staining, which stains a particular matrix structure called the Peri neuronal net. And that is a net of matrix that's CSPG rich that surrounds neurons in the nervous system. And it's stained, and you can see it really forms a net right around the neuron. And during development, what happens is that initially there's no perineuronal net. And what happens during development, connections are made. Let's consider this neuron here. Connections are made, and those connections, when they're made, they stimulate the neuron and the surrounding glia to make this perineuronal net that is CSPG rich. And what that does is it sort of cements in the connection. It stabilizes the connection so that no new things can come in, and these axonal ends do not sprout away. It stabilizes the connection. When there's been an injury, there may be a stimulus for a spared axon, consider this a spared axon for a moment, to synapse on a deafferented neuron. But that may require, that sort of remodeling may require a decrease in the CSPG in the perineuronal net. And we believe that what's happening in the Sox9 knockouts is we're opening up that window for neuroplasticity because we're decreasing the CSPG all through the neuroaxis. And in fact, when we look for WFA staining in the knockouts, you can see that there's a reduction, a great reduction, far away from the lesion in this WFA staining in the perineuronal net. So that's what we think is happening. We think that the Sox9s do better, the knockouts do better, perhaps because there's growth through the lesion, though we have no proof of it. But perhaps also what we're doing is we're just increasing the window for neuroplasticity above and below the lesion. And that allows for this rewiring of circuits that can underlie improved behavioral outcomes. How am I doing? I'm doing okay. Then I want to just show you this for a second. Obviously, the first set of experiments I showed you don't really answer the more clinical question as to whether or not reducing Sox9 after injury would be beneficial, because we really knocked out Sox9 ahead of the injury. But what we've done here is we've done a spinal cord injury, a contusion injury, waited a week, and then we do tamoxifen administration. we calculate that that will result in a knockout of Sox9 somewhere around 10 to 14 days after the injury, because the process takes a little time. And then again, we looked for behavioral outcomes. And on the open field test, again, so they're indistinguishable initially because the knockout really hasn't occurred yet. We give it tamoxifen at 1 week, and eventually the knockouts do better in the open field testing than the controls. And once again, if you look at the activity boxes, the Sox9 knockouts by 14 weeks after the injury are indistinguishable in terms of the amount of movement they do, their mobility in these activity boxes relative to uninjured animals, and they do about 2 or 3 times better than the injured controls. Now I just want to switch gears. This is all about spinal cord injury, but I just want to show you some data that I think speaks to the underlying mechanism, and when you address a mechanism that is basic enough, it may actually extend to other areas of CNS disease. So we reasoned that if neuroplasticity underlies the improvement in the Sox9 knockouts, that other conditions of the nervous system in which neuroplasticity is important for recovery, they may also do better after in the Sox9 knockouts. So stroke is a good example of that. Human and animal studies suggest that after stroke, there is a period of time after which people get better, and animals do better. And it plateaus in humans sometime between 3 months and 6 months. Whatever benefits are gained, it sort of plateaus. And a lot of the evidence suggests that the reason why there is this improvement in neurological function is because there is this neuroplasticity. The neurons that are not damaged by the stroke are able to subsume the function of neurons that were lost due to the stroke. And so if that's due to structural neuroplasticity, we figure that the Sox9 conditional knockouts should do better even in this model. So it wouldn't be specific to spinal cord injury. So I just want to show you this for a bit. This is doing MCAO, middle cerebral artery occlusion. This is just some staining at 24 hours post-injury. The white area shows the area of the lesion in controls and in knockouts. And we just want to confirm that the lesions are the same in both types of animals. This is looking at Sox9 expression. So again, in stroke, so these are the controls. And you can see this is the contralateral side to the injury. It was a left-sided stroke. You can see there's a great increase in Sox9 on the stroked side, the left side. That's the green staining, lots of Sox9 positive nuclei, lots of increase in GFAP. And look at the knockouts. Again, as expected, there is a great reduction in Sox9 expression and correspondingly a reduction in GFAP expression, going along well with the expression data I showed you. What happens behaviorally? I don't want to do this in too great detail, but these are 3 different tests of behavioral recovery, really of left-right asymmetries. So this is a corner test in which you allow the animal to enter a corner, it rears, and it turns left or right. And typically after the stroke, they'll turn left. And you can see that the controls turn preferentially left for a long time after the injury. They get 10 trials. Whereas the knockouts very early on return back to the baseline of 50-50, left and right. This is a cylinder test, really testing whether the animal explores its environment with the right paw, which is the injured side, or the left. And you can see that the knockouts return to a symmetrical use of right and left paws much earlier than the controls. And this is just looking at return of grip strength. And again, here the Sox9 knockouts are on top. And they recover grip strength much quicker in the affected paw. So they do better even in this model of injury. This is looking at WFA staining. If we get the lights once more, I'd appreciate it. Whoever magically did that before. So WFA is the red staining. These are the controls. SMI32 is for certain neurons in the red nucleus that project down the spinal cord. And you can see they have nice red WFA staining surrounding it. And it's really absent in the knockouts, or largely absent. And so we think, again, that what we're doing is we're opening up this plasticity, this possibility of plasticity that will underlies even an MCAO recovery. Thank you. And this is just looking at CSPG expression instead of WFA, again, in the controls or the knockouts. In the last few minutes, that's going good. In the last few minutes, I want to talk about how can we move this, or how are we moving this towards pharmacological use of this information. So what we've done is we're basing this about what we know for Sox9 function. Sox9 as a transcription factor, it's synthesized in the nucleus, and synthesized in the cytoplasm. It has to get to the nucleus to affect gene transcription, gene expression. And it does so by binding to calcium binding protein calmodulin, and then it gets transported into the nucleus. And so it occurred to us that one way to affect Sox9 activity would be to block calmodulin. And there are many different calmodulin antagonists. Calmodulin is a dumbbell-shaped protein, and these antagonists typically bind in the middle of the dumbbell and cause it to collapse, so it can't interact with its partners. And so we tried various calmodulin antagonists. Almost all of them work and do what I'm going to show you here. This is 2, CSA and CPZ. And you can see that in our astrocyte cultures, these drugs turn down GFAP, xylosyltransferase 1, collagen, aggrecan, brevican. So there's pharmacological ways to get at Sox9 activity. This is now showing you 2-APB. 2-APB is a drug that blocks TRPV calcium channels for internal calcium stores, and that would obviously interfere with calmodulin function. And you can see again, we get this reduction in Sox9 target gene expression. So one way to get at this is through calmodulin. I've shown you some drugs. We've also identified a peptide. So a peptide strategy. So this is the sequence here shown in gray in Sox9 and various other Soxes. You can see it's well conserved. This is the peptide that encodes the calmodulin binding sequence. And so what we've done is we've synthesized that peptide as a TAT fusion protein. And the idea is that if you can flood the system with the peptide, that you'll compete out for binding to calmodulin and reduce Sox9 gene expression, Sox9 target gene expression. And so when we go to an astrocyte culture system and we use the TAT fusion peptide, we're able to reduce all these Sox9 target genes quite nicely. Okay, so there's a peptide strategy here. Another completely different strategy is based on our knowledge that Sox9 has to dimerize in order to turn on genes that are involved in chondrogenesis. That's the matrix genes, okay, collagen, et cetera. And so we've designed a way of interfering with the dimerization of Sox9. And when you do that, again, and use our astrocyte culture system, you reduce the GFAP, you reduce the Sox9 target genes, including link protein and collagen 2A, and some of the core proteins for CSPGs. So there are different strategies, and it would be wonderful to be able to show you that these pharmacological methods of blocking Sox9 actually improve recovery in spinal cord injured animals. I can't show you that yet today, we're not quite there. But what I'll show you is our first in vivo experiments seeing whether or not any of these pharmacological methods will reduce Sox9 target gene in the spinal cord injured animal. And this is showing you expression of the Sox9 genes, target genes like GFAP and collagens, aggrecan, brevican, neurocan, in 4 different doses of CPZ. And this is done in the spinal cord injured animal. So the animal gets an injury, and then intrathecally we deliver the drug for 1 week, sacrifice the animal, and then we do real-time PCR. And you can see there's a real good effect, giving us a feeling of a dose response here, the highest concentrations being the most successful at reducing Sox9 target gene expression. So the next step that we are on is to try and see whether or not this results in better neurological recovery, in reductions in CSPGs, reduction in perineuronal net. And the final slide that I'll show you, pretty good, is that this is not just a phenomenon in the rat or the mouse. Thanks, Mike. This is, these are pathological samples that we got from the pathology department here at the university, at the UH. looking in human samples. So this is a section from a person who passed away many years after spinal cord injury. Sox9 immunostaining in red and GFAP in green. And you can see at the site of the lesion, there's lots of Sox9 expression and lots of GFAP, of course. In traumatic brain injury, you see increased Sox9 expression and, of course, the increased GFAP. Increased Sox9 expression after hemorrhagic or ischemic stroke, and even around a tau-positive plaque in a patient that had a diagnosis of Alzheimer's disease, you can see Sox9 expression in cells surrounding the plaque. So what we think is happening is that Sox9 is really part of a generalized program. When the nervous system is damaged or injured in some way, is inflammation, this turns on Sox9, and Sox9 activity is going to result in increased astrocyte activation, increased CSPG production, perhaps throughout the nervous system. And when you're able to inhibit Sox9 activity or expression, you may reduce at the lesion sites and elsewhere CSPGs, and that can open up this neuroplasticity and perhaps affect better repair. Just for a moment, we'll thank the folks who do the work. So first of all, this is the spinal cord injury group at Robarts. Really indebted to Lynn Weaver, who got me started in spinal cord injury. Greg Decaban, he gives the group its immunology flavor and is an immunologist. Paula Foster allows us to do small animal imaging and see what's going on in the cords of these animals. This is a relatively recent picture from my lab, Feng, and this is Kathy. Kathy did all the MCL work I showed you. This is Vanessa, who helped us with the animals. Monty is a doctoral student in the lab who did the knockout studies that I showed you. Nicole and Todd do a lot of the in vivo work that I've shown you, and Todd specifically has been really key to looking at the Sox9 inhibitors. These are our funding partners, and I thank you and take questions.
Arthur, do you have a beautiful talk? Do you have any information on sensory systems? So either positive
or negative. Yeah, it would be great. So that's the, as me and Lynn wrote, the dark side of neuroplasticity. If you have too much neuroplasticity or inappropriate neuroplasticity, you can actually rewire the cord to result in autonomic problems or pain problems. I'm really waiting, hoping to be able to do pain studies in rats when I get a good Sox9 inhibitor, because it's, at least in our hands, very difficult to do reliable pain studies in mice. But we are working towards doing autonomics in the mice. That will give us a clue as to whether or not there is some of this negative. One of the reasons why I'm hopeful that we won't get that, we need to test for it, is that because I think that the behavioral outcomes are better because of short-range rewiring. And so who's most likely to sprout in the region of a deafferent target, but it's something that's already targeting that same pool of neurons. So I'm hopeful that it's going to be okay, because if you look at the behavioral outcomes, they're much better really quick after injury. So I'm hoping that we won't get some of those negative types of neuroplasticity, but it's definitely something we need to test.
Go ahead, Dr.
Session.
Hi. So I wanted to talk a question again with this maladaptive plasticity. Did you note in any of these cores that you looked at there was more sprouting of any fiber types?
We looked for serotonin below the lesion. And there is increased serotonin below the lesion, which we think underlies potentially the motor improvements. The animals have no obvious problem. So they have no obvious, no phenotype. There's no ouchiness. They don't seem to, in fact, if you look at the locomotor box behavior, they seem to be really bright and really active, which is sort of opposite to what you'd think if they were, for example, in pain. But you really need to test for the neuropathic pains and the others with something more specific. It's just that in the mice, we find that difficult because mice are more skittish.
So at basal levels, if you do a CSPG staining before even you do the injury, is there differences in
I will say it depends on how close to the knockout you do it. I think if you looked several weeks after knocking it out with now no injury, you will see a drop in CSPGs because it has to be turned over.
foresee a certain time period that you'd have to administer a Sox9 inhibitor for plasticity?
Yeah, great question. So it's something we would like to do again. So right now, the longest we've gone is we've waited a week, and then we give tamoxifen, which we, based on various studies, we think effectively gives us knockout somewhere around 10 days to 2 weeks after the injury. What we would love to do is do a tamoxifen administration, say 4 weeks or 6 weeks after the injury. My feeling is just like any other protein, it needs to turn over. And the behavioral, so the question would be, do other things limit neuroplasticity? And probably the answer is yes. Other than the CSPGs, can we still get some benefit? Our 1 week data that I showed you makes me hopeful.
So one last question. So great talk, Arthur. Just finds the corollary of that question is actually to the earlier one. So if you have to have a turnover of CSPGs, what is their pathway? And it's suggesting if you're having an injury, you have your inflammatory response, everything we know about that, and if you can wait almost 2 weeks before modulating CSPGs and still see an effect, what does that tell you about how early you truly need to be then? Can you come earlier in and see more profound effect?
Maybe. So if you take a look, if you compare our Sox9 knockouts done before the injury, and I have to say the reason why we did that is because we didn't want tamoxifen as a confounding issue on board. So we gave tamoxifen for 1 week, then we allowed a 1 week washout, and then we looked at it. And those animals, so they actually, if you were to plot all 3 groups, those that had the knockout before injury, those that had no knockout, and those that had knocked out 1 week after, you'd find that you get the best recovery when you knocked it out before, and you get the ones that had knockout 1 week after kind of lie in the middle. They do better than controls, but not quite as good as the ones that started with the knockout earlier. So I think there is some benefit. It might be that, there's a huge increase in Sox9 and all these genes right after injury, and it may be beneficial to block that initial one, which we haven't done in the later knockout. So we may miss that opportunity. Okay.
So we'll move on. Thank you, Art.
Thank you.